If you watched my last video on the HPV vaccine and caught yourself nodding along when I said non-inferior, but then quietly Googled it afterwards, this one's for you. No shame. Non-inferiority trials are probably the most widely misread study design in clinical medicine, and that's not an accident. Let's fix that in about 3 minutes. So, what is a non-inferiority trial? A standard randomized trial asks, is the new thing better than the old thing? That's superiority. Straightforward. A non-inferiority trial asks a different question. Is the new thing good enough compared to the old thing? Not better, just not unacceptably worse. The key word is unacceptably, because not unacceptably worse is doing a lot of work in that sentence, and whoever defines unacceptable has a lot of power. That threshold gets a name, the non-inferiority margin. It is set before the trial starts, and it represents the maximum difference investigators are willing to tolerate. If the new treatment stays inside that margin, even if it's technically a little worse, the trial declares non-inferiority. So when does it make sense to do a non-inferiority trial? Here's where it's actually the right call. First, when a placebo control is unethical. You can't withhold a proven therapy just to run an experiment. In the HPV trial, investigators couldn't randomize kids to no HPV vaccine, so they asked the question, is one dose no worse than 2? That's an ethically sound question with a practically important answer. Second, when the new treatment offers a real-world advantage, like fewer doses or better tolerability, and you want to know if it's close enough to justify the trade-off, NI is for you. One HPV shot that works like 2, that's a public health home run, especially in low-resource settings. This design exists for a reason. It answers questions that superiority trials can't. Now, here's where it gets interesting. Non-inferiority trials are a tool that pharmaceutical industries can use because they're dramatically easier to pass. Think about it. You don't have to prove that your drug is better. You just have to prove it's not worse than the other drug. And here's the trick. You might get to help pick the margin. A sponsor can choose a generous margin with a small enough sample size and a surrogate endpoint that can't fail.
And using this, you cannot fail.
Watch for these red flags. A margin that seems arbitrary or wide. A surrogate outcome instead of a patient-oriented one. An active comparator that's underdosed, that makes the new drug look better.
Maybe dressed up. I think you should dress up too.
And industry funding with no independent adjudication. Non-inferiority trials aren't inherently corrupt, but they could be gamed. And knowing the difference between a well-designed one and one that's dressed as a statistically significant one is a whole ballgame.
And that is half the battle!
There are a few possible outcomes when you're interpreting a non-inferiority trial. Let's take a look at this graphic. The x-axis shows the rate difference between the new treatment and the standard. 0 means identical. Move to the right, the old treatment is winning, and move to the left, and the new treatment is winning. The dashed vertical line on the right is the non-inferiority margin. In the HPV trial, that's 1.25 infections per 100 patients. Every confidence interval you see in a non-inferiority trial falls into one of these 3 zones. The top band is inferior. The confidence interval crosses the margin, or it sits entirely beyond it. You cannot rule out that the new treatment is unacceptably worse. Trial fails. The middle band is non-inferior. The entire confidence interval, including the upper bound, sits below the margin. The new treatment might be a little worse, it might be identical, it might even be better. But you've ruled out unacceptably worse. Trial passes. The bottom band is superior. The entire confidence interval sits to the left of 0. The new treatment is statistically better than the standard. That's a bonus. Non-inferiority trials aren't designed to show this, but it happens. The key move. Watch the upper bound of the confidence interval. That's the only number that determines pass or fail. So let's apply this with some real-world numbers. If you remember the HPV trial, from the nonavalent vaccine, that's Gardasil 9, 1 dose versus 2 doses gave a rate difference of 0.21 infections per 100 participants. So 1 dose had marginally more infections than 2. The 95% confidence interval ran from negative 0.09 to 0.51. The upper bound, 0.51. Margin, 1.25. It's not even close. The confidence interval sits deep in the non-inferiority zone, nowhere near the margin. Non-inferiority declared. And here's the thing. The point estimate of 0.21 still technically favors 2 doses. So don't go calling this equivalence. Non-inferiority means not unacceptably worse than. That's a precise, intentional claim. Don't stretch it. So in conclusion, in the HPV trial, the margin was pre-specified at 1.25 infections per 100 participants, which was back calculated to guarantee single-dose efficacy above 80%. That's rigorous and how it should be done. This one holds up. That's what a clean non-inferiority trial looks like. Now you'll know when you're reading one and when someone's trying to sneak one past you. Until the next time, this is Dispense is Written, and I'm Greg.